Hello everyone. I'm Salil, assistant professor from the Department of Pharmacognosy, Sri Shanmuga College of Pharmacy. Today I'm going to talk about the one of the foundational concept in the pharmacognosy. So, evaluation of a crude drugs. Let's get. First of all, evaluation of crude drugs. What is meant by evaluation of a crude drug? Why do we need? Okay. Evaluation of crude drug means ensuring the identity, quality, and the purity of the drug before it reaches the patients. Okay. Now, why do we need to evaluate the crude drugs? So there are some three major reasons behind the evaluation of crude drugs. One is a biochemical variation in the drug, and also on effect of a treatment and storage of a drug, and adulteration and substitution. Okay, so biochemical variation means the plant can have a different chemical composition depending on where and also when it's grown. Okay, so, an effect of treatment and storage means improper drying, storage or processing can degrade the quality of the crude drug, and also adulteration and substitution. Okay, so it means inferior or fake materials are sometimes deliberately or accidentally mixed with the crude drugs, so which can directly affect the quality of the crude drug. Okay, so evaluation of a crude drug is kind of our quality check. Okay, so first is about the organoleptic evaluation. Okay, organoleptic evaluation. The first and simplest method is organoleptic evaluation using our five senses. Okay, so this is a qualitative method based on the morphological and also sensory characters. Okay, so we study the form of drug. It means that's called the morphology, and we describe the form means it is called the morphography. Okay, so what do we observe in the organoleptic evaluation? So color, odor, taste, shape, size, and sometimes some special features like touch and textures and a fractured surface and some general appearances. So these are like a quick and no equipment observations that gives our first clue about the quality of a drug, quality of the crude drug. Okay, if is there any problem with this natural sensory characters, it means we can directly find out that the quality of the crude drug was affected. Okay, so next is about the microscopical evaluation. Okay, so when organoleptic evaluation is not enough to give a enough, we will go deeper. Okay, so that is microscopic evaluation. So this is done to confirm the structural details of the plant origin drugs. So we can examine the drug either in its original form or in a powder form using the transverse or longitudinal sections. Okay, so examples of some microscopic characters are lignified trichomes of the Naxomica and the wavy medullary rays of Cascara bark. Okay, so we also have a chemomicroscopy where we apply the chemical reagents to sections or powder to identify the specific constituents. Okay, so fluoroglucinol with HCl can visualize the lignified cells in blue color, and also ruthenium red ruthenium red will visualize the mucilage in a pink color, and also iodine plus starch gives you a blue color. So this helps us chemically confirm what we are seeing under the microscope. Okay, so next is about the leaf constants. Okay, so still under the microscopy we have some important parameters called the leaf constants. So these are like a numerical values specific to the each plant species. Okay, so those parameters are stomatal number and stomatal index and vein islet number and vein termination number and palisade ratio. Okay, so stomatal number means average tomato per square millimeter of epidermis, and stomatal index means percentage of stomato among the total epidermal cells, and vein islet number means number of vein islets per square millimeter midway between mid ribbon margin, and vein termination number means the number of vein endings per square millimeter. Okay, so palisade ratio means average palisade cell beneath the each epidermal cells. Okay, so these values are like a fingerprint for each plant, and also it is very useful in identifying the adulterated leaf drugs. Okay, so next is about this stomata. Okay, so let me briefly explain the stomata since we have mentioned in the stomata index. So stomata is a minute opening on aerial plant pods. Okay, so and it has a central bore and two kidney-shaped guard cells, and also it's surrounding by the subsidiary cells. Its primary function of the stomata is gaseous exchange, and also secondary function is about the transpiration. Okay, so there are five types of stomata. Is there? One is a those are all parasitic, diacetic, and anisocytic, anamocytic, and actinocytic. Okay, so each type is a diagnostic for specific plant families. Okay, so next is about the trichomes. Okay, another microscopic characters trichomes are a plant has. Okay, so these are like a tubular or a glandular outgrowths from the epidermal cells. They may not always have a direct functions, but some perform secretory functions. Okay, so types of trichomes are non-glandular trichomes. They are protective, and also glandular trichomes. They are like secrete oils or resins, and also hydrathodes. Hydathode means a special types of trichomes involved in the water secretion. Okay, so trichomes shape and structure are highly diagnostic for drug identification. Okay, so next is about the under microscopy. We also look at the cell contents. Okay, things like starch grains and alurin grains and mucilage, tannins, oil globules, and inorganic matters like calcium carbonate and silica. Okay, so calcium oxalate crystals are especially important. They are appear in the different forms. Okay, like a cubical or prisms or a rhombic or a tetragonal, monoclinic, and acicular. Acicular means a needle-like structure and rosettes. So each drug has a characteristic crystal type which helps in the identification of crude drug. So now chemical evaluation. So this covers the isolation, purification, and identification of active constituents. So we have two types. One is a qualitative identification and a detection of adulteration. So quality of adulteration means qualitative test or identification test means chemical test for each secondary metabolites. So therapeutic value, active constituents of the specific root drug is the one responsible have the responsibility for the medicinal value of the plant, so that we can. So the therapeutic value is mostly depends on the quality of the secondary metabolites, so that we have to identify the quality and also quantity of the secondary metabolite. So for this we have some identification tests for specific secondary metabolites like alkalides, that is for alkalides, test for tannins, test for glycosides, and test for resins, and also test for terpenoids, volatile oils. Okay, so these tests are quick chemical reaction that confirm the both identity and also purity. Okay, so next is about the physical evaluation. Okay, so physical evaluation uses the physical standards, uh, though these are not always constant and are mostly applied to the unorganized drugs like resins, oils, and waxes. So some key parameters includes moisture content, viscosity, melting point, solubility, optical rotation, refractive index, ash values, extractive values, volatile oil content, and foreign organic matter, and finally chromatography and also spectroscopy. Okay, so these give us measurable and objective data about the crude drug. Okay, so next is about the biological evaluation. The most advanced method the biological evolution or a bioassay. So when we want to confirm the therapeutic activity of the crude drug, we test is we test the crude drug on the living organism. This is called the bioassay. So measuring how much of sample is needed to produce the same biological effect as a standard preparation. Okay, so this activity is expressed in international units. IU, for example, one international unit of a digital is equal to activity in the 76 milligram of standard preparations. Okay, so there are three type of bioassay method is there. One is a toxic method and the symptomatic method and the tissue method. Toxic method means whole animal is used. Symptomatic method is whole animal is used. In tissue method, isolated organ or tissues prepared culture is used. So some specific bioassay examples are mice. Mice are commonly used for the toxicity studies to find out the lethal dose and the effective dose, and pigeons used to assay the digitalis glycosides allergen injection at a rabbit's eye for drug affecting the eye, and also coxcomb or a rabbit uterus to study the effects of a gut and human beings is the next level of studies. Then microbiological assay using the bacteria, yeast, and molds to antibacterial molds to test the antibacterial drugs and assay, and also some vitamins. So bioassay gives us a most clinically relevant information about the drug. Okay. So to summarize, the evolution of a crude drug involves the five key methods: organoleptic methods and organoleptic evolution, microscopic evolution, chemical evolution, physical evolution, and biological evolution. So each method builds on the previous and giving us a complete picture of the drug's identity, quality, and also the potency. Okay, so thank you all for the attention, and I'm happy to take this session. So thank you.